Hello students, welcome to this session. I, Dr. Rajeshri Mutha, MBBS DPB from CPS, College of Physician and Surgeon, Anauti Hospital, going to take lecture on general microbiology. Let's study some scientists in microbiology. So first is Anthony van Leeuwenhoek, first scientist to observe bacteria and other microorganisms using single lens microscope. Louis Pasteur, father of microbiology, introduced sterilization techniques, described pasteurization of milk, vaccine development against anthrax, fowl, cholera, and rabies. The Robert Koch discovered anthrax bacilli, tubercle bacilli, cholera bacilli, and Koch's postulates. So in Koch's postulates, first is the microorganism constantly associated with the lesion of the disease. Second, isolate the organism in pure culture. Third, same disease must result when isolated organism is inoculated into suitable laboratory animal. Fourth, possible to re-isolate the organism in pure culture. Fifth, antibody to causative organism should be demonstrable in patient serum. Edward Jenner developed first vaccine, smallpox vaccine. Joseph Lister, father of antiseptic surgery.
Hello students, today we will see the introduction of microbiology. So what is microbiology? It is study of microorganisms like bacteria, virus, fungi, parasites. It is called as microbiology. So first is scope or application of microbiology. So first is in microbiology there is identification of microorganisms like bacteria, virus, fungus and parasites, and then report of sensitivity is done to further treatment of the patient, and some serological reaction studies is also done for fast diagnosis of the disease, that is called as indirect diagnosis of the microorganism. So examples of that is rheumatoid arthritis test for rheumatoid arthritis, ASO test for rheumatic fever, CRP test for inflammatory disease, HBsAg test for hepatitis B, venereal disease research laboratory test for syphilis, HIV human immunodeficiency virus, for AIDS acquired immunodeficiency syndrome, HCG test for pregnancy, Vidal test for typhoid fever. So these are few serological reactions for the diagnosis of the disease and then treatment of the disease. Thank you.
Hello students, let's study sterilization and disinfection. Sterilization process by which all living microorganisms, including viable spores, are either destroyed or removed from an article or from the surface medium, results in the reduction in more than 10 to 6 logs colony forming units of microorganisms and their spores. Disinfection process that destroys or removes most of, if not all, pathogenic organisms, but may not destroy bacterial spores. So result is reduction in 10 raised to 3 log CFU in most organisms and not spores, achieved by physical agent or chemical agent, usually used only in inanimate object, not on body surfaces. Factors for efficacy of sterilant or disinfectant: organism load, nature of organism, concentration of the disinfectant, contact time, temperature stability and relative humidity, biofilm which prevents the entry of disinfectant. Or property of ideal disinfectant is the broader microcidal activity, fast acting, not affected by environmental factors such as organic matter, non-toxic, compatible with surfaces, odorless or pleasant odor, economical and environmental friendly. The sterilant, which kills all bacterial spores, tubercle bacilli, non-enveloped viruses, fungi, vegetative bacteria, enveloped viruses. So examples are the physical methods: steam sterilizer, that is autoclave; dry heat sterilizer, that is hot air oven; filtration; radiation, ionizing and non-ionizing; others like incineration and microwave. In chemical methods, ethylene oxide sterilizer, plasma sterilizer. So Spalding classification for medical devices. So critical device, that is high risk. So items that enter normally sterile sites. So recommended method is sterilization, and the medical instruments like surgical instruments, implants, prosthesis, rigid endoscopes, syringes, needles. Semi-critical device, intermediate risk. Items in contact with mucous membrane or body fluid. Recommended method is disinfection, high-level disinfectants. Examples are respiratory equipment, non-invasive flexible endoscopes, bedpans, urine bottles. non-critical low risk item in contact with intact skin, disinfection, intermediate or low level disinfecting. The examples are non-critical patient items, non-critical environmental surfaces. Hello students, let's study moist heat. So in that we will see autoclave in detail. So it also called as steam sterilizer. The principle, when water is heated in closed container, it produces vapor and then it causes vapor pressure. Then when water boils in closed container, the vapor pressure is increased more than the atmospheric pressure, and then it has a more penetrating power to kill the organisms. And the mechanism, killing of organisms by irreversible coagulation, denaturation of enzymes and proteins. The types of autoclave are horizontal when it is in large volume, and it is mainly used in CSSD department. And the vertical one is in small volume, used in most laboratories. The components, there are three main components: pressure chamber, lid and electrical heater. So pressure chamber is consists of the large cylinder, either vertical or horizontal. It is made up of metal or stainless steel, and it has a steam jacket, that is water compartment at the bottom. The lid, it has discharge tap for passage of air and steam, pressure gauge for the pressure measurement, and safety valve to remove the excess steam. An electrical heater, it is attached to the jacket that heats the water to produce steam. The working procedure: the materials to be sterilized are placed inside the cylinder. A steam jacket is filled with sufficient water, and then lid is closed with the help of clamps, and the electrical heater is put on. Now there are three phases. So first is conditioning phase, second is exposure phase, and third is the exhaust phase. So in conditioning phase, you have to give condition of temperature, pressure and time. That is temperature 121 degree Celsius, pressure 15 lbs and time 15 minutes. So after the water boils, the air in the chamber is completely displaced by the steam. So the steam pressure rises and then it reaches to the desired level, that is 15 lbs pressure. Then the safety valve opens and excess steam is removed out. Exposure phase, the holding period is for 15 minutes. Exhaust phase, so after the holding period is over, the electrical heater is switched off and the steam sterilizer is allowed to cool till it reaches the atmospheric pressure, and then the material inside is removed. The care and maintenance of the autoclave. So we have to always put water in steam jacket. Do not forget to put off gas burner after the holding period is over. Never leave autoclave unattended when pressure is rising, and never open the lid immediately after heating period is over. So we have to allow it to cool and then open the lid. Sterilization control, physical conditions like temperature, time and pressure, chemical indicators like external pack control, that is autoclave tape, or Browey test or internal pack. The biological indicator, spores of Geobacillus steothermophilus are the best indicator which kill spores, in which the spores are killed in 12 minutes at 21 degrees Celsius. Uses of autoclave. So there are mainly two uses: hospital uses and the laboratory uses. In hospital uses, the sterilization of surgical instruments, anesthetic equipments, dental instruments, implanted medical devices, surgical drapes and linen, biomedical waste treatment for disposal. Laboratory uses, sterilization of glassware, test tubes, slides, pipettes, petri dishes used in the laboratory, sterilization of culture media for preparation, inoculated infected culture plates, killing of microorganisms and the spores. Flash sterilization, it is modification of conventional steam sterilization used for the emergency or during unplanned procedure. So it involves fast sterilization, that is temperature 134 degrees Celsius for 3 to 10 minutes. For surgical instruments in unwrapped condition in steam sterilizer located close to the operation theatre, and it is restricted for emergency situation. Disadvantage: not suitable for porous or cannulated instrument or implants because the instruments are not packed, so they remain wet following sterilization, and there is high risk of recontamination. Right. So in that, examples are flaming, holding instruments to be sterilized in flame of the spirit lamp or Bunsen burner. Example, wire loop, points of the forceps, spatula, scalpel, needle, mouths of culture tubes, glass slides. Second method is incineration. It is physical method of sterilization in which material to be disposed of are kept in big dig and are burnt off. It is called incineration. It burns, that is, sterilizes the anatomical waste and microbiology waste by providing very high temperature, 800 to 1200 degrees Celsius, converting the waste into ash, fuel, gas, and heat. Uses in this is excellent method for rapidly disinfectant material such as soil dressing, infected dressing, bedding, pathological material, disposable syringes, gloves, hospital waste, lab waste, etc. Disadvantage, plastic material emits clouds of the black. The third example in dry heat is the hot air oven principle. When electricity is passed through heating coils, electrical energy is converted into heat energy. The temperature is 160 degrees Celsius and holding time is 2 hours. Uses dry sterilization of syringes, needles, drying of glass waste, test tubes, petri dish slides, pipettes, heating of chemicals used for preparation of standards, sterilization of oil, grease, liquid paraffin. Sterilization control of the hot air oven, biological indicator, the spores of Bacillus atrophius. The uses of hot air oven, sterilization of egg media, sterilization of serum media, sterilization of sugar media. Filtration. So filtration acts by removing microorganisms, not by killing. There are two types of filters. Depth filter used as drinking water purifier. Membrane filter widely used in hospital. Bacterial filter size is pore size is 0.22 micrometer. Filtration of air, surgical, three-ply mask and respirators. HEPA filters, high efficiency particulate air filter, removes 99.7% of particles that have size of zero point 0.3 micrometer or more. ULPA filters, ultra low particulate penetration air, removes 99.999 particles that have size of 0.12 micrometer or larger. Filtration of liquid used for bacteriological examination of water in hospital settings, dialysis water, to remove bacteria from pharmaceutical fluids that are heat labile and cannot be purified by any other means. Radiation, ionizing and non-ionizing radiation. So ionizing radiation include cobalt 60 gamma rays or electron accelerators. Use low temperature sterilization method that has been used for a number of medical products, tissue for transplantation, pharmaceuticals, medical devices. Mechanism causes ionization of molecules in organisms leading to breakage of DNA. Advantage, high penetrating power and rapidly acting. Temperature is not raised, so it is also called cold sterilization. Disadvantage, high sterilization cost and may have deleterious effect on equipment made up of polyethylene. Sterilization control, efficacy of ionizing radiation checked by Bacillus pumilus. Non-ionizing radiation, infrared radiation technology, alternate method of sterilization for selected heat-resistant instrument. Ultraviolet radiation does not achieve sterilization described under intermediate level disinfectant. Test for sterilizers, the indicators, physical indicator, time, temperature, pressure. Chemical indicator, they use heat or chemical sensitive material which undergo color change if the sterilization parameter, that is time, steam, quality, temperature for which it is issued is achieved. Common types are class 1, class 2, class 4 and class 5. Biological indicators, most reliable indicator as it uses bacterial spores to check the effectiveness of sterilization. Spores are highly resistant and will be destroyed only when the effective condition is achieved. Geobacillus stereothermophilus for heat sterilizer, gas plasma hydrogen peroxide and liquid acetic acid sterilizer.
Disinfection is the process that destroys or removes most of the pathogenic organisms but may or may not be the spores. So it is achieved by physical or chemical agents. So classification of chemical agents. So first type is alcohol group, That is examples: ethyl alcohol, isopropyl alcohol, aldehyde, formaldehyde, glutaraldehyde, halogens, iodine, chlorine, phenol, cresol, lysol, chlorhexidine, oxidizing agents, hydrogen peroxide, dyes may be malachite green, crystal violet, metallic salts of silver, copper, mercury, gas sterilization by ETO, ethylene oxide, or plasma sterilization. Now let's see the level of sterilant or disinfectants according to their microbiological actions. So these are the 5 factors for the microbiological actions on vegetative bacteria, bacterial spores, tubercle bacilli, fungi, enveloped viruses and non-enveloped viruses. So sterilant is kills all the forms of the microorganisms. Then in disinfectants there are 3 types: high level, intermediate level and low level. So high level disinfectants kills all the morphological forms of the organisms except bacterial spores. Intermediate level do not kill or destroy bacterial spores, but they can damage all the microbiological forms. Low level, they do not kill bacterial spores and fungi plus minus, non-enveloped viruses plus minus. Factors influencing the efficacy of the disinfectant. So it is organism load, nature of the organism. So example, prions or bacterial spores are highly resistant than the other microorganisms like mycobacteria, viruses, fungi or vegetative bacteria. Then concentration. So optimal concentration is required. Then contact time, temperature, stability, relative humidity, biofilm formation which prevents entry of disinfectant. So these are all the factors. An ideal disinfectant should be broader microcidal activity, fast acting, not affected by the organic matter, non-toxic, then compatible with the material, odorless and economical. The antiseptics is a type of disinfectant which is safe to apply on the body surface, that is skin and mucosa, which results in the destruction of the organisms. Then cleaning or decontamination is the reduction in the pathogenic microbial population to a level at which items are considered as safe without protective attire. Spaulding's classification of medical devices. So they are categorized into the critical device, high risk, semi-critical device, that is intermediate risk, and non-critical device, that is low risk. So critical device is item that enter a normally sterile site. So they are invasive. The examples are surgical instruments, implants, prostheses, rigid endoscopes, syringes, needles. So the recommended method is sterilization. Semi-critical device, they are in contact with the mucous membrane or body fluids, but they do not enter. So they are respiratory equipments, non-invasive flexible endoscopes, bed pans, urine bottles. So the recommended method is disinfection, that is high level disinfectants. Non-critical or low risk devices, they are in contact with the intact skin. So non-critical patient items like an environmental surfaces. So the method of recommendation is the disinfection, that is intermediate level or low level disinfection. Agents used in the hospital for achieving sterilization, disinfection and cleaning. So they are divided into the high level, intermediate level, low level and agents of the cleaning. So high level disinfectants, the examples are aldehydes, in that formaldehyde and glutaraldehyde, parasitic acid and hydrogen peroxide. Intermediate level disinfectants are alcohols, example ethyl alcohol, isopropyl alcohol, phenols, then cresol, lysol, halogens, iodine and chlorine. Low level disinfectants, quaternary ammonium compounds and chlorhexidine. Agents of cleaning are enzymatic solution, detergent, soap, either antimicrobial or plain soap. So let's see the disinfectant. So first is high level disinfectant, so capable of killing all the microorganisms plus bacterial spores. So first is the aldehyde. Commonly used are formaldehyde, glutaraldehyde. So mechanism is combine with nucleic acids, proteins and inactivate them. So the formaldehyde is excellent high level disinfectant. Uses, it is largely restricted to non-patient care area for preservation of anatomical specimens, histopathological specimens and stool specimen. The fumigation of OT is nowadays practice not used. Disadvantages: it is irritating and pungent odor and potential carcinogen, corrosive, causes skin irritation and asthma when inhaled. Glutaraldehyde, so it is used for the semi-critical items like endoscopes, cystoscopes. So it is also used for aerial disinfection and cleaning, that is fogging and cleaning of floor and surfaces of the critical areas. So example, bacillocid is used in the concentration 2%, it is example Sidex. Duration is 20 minutes and for spores is 10 to 14 hours. Advantages: it is active in the organic matter. Disadvantages: pungent odor, can produce eye irritation and asthma and contact dermatitis. H2O2, hydrogen peroxide, mechanism of action: it produces hydroxyl free radicals which are toxic and attack the various cell components. The uses are sporicidal, more than the 4 to 5% concentration. 3% H2O2 for the environmental surface disinfection, fogging and wound cleaning. 3 to 6% H2O2 for disinfection of the soft contact lenses, ventilator, endoscopes. Say 6 to 7.5% H2O2 as a chemical sterilant in plasma sterilization. Vaporized H2O2 used in the industrial sterilization of medical devices and for decontamination of the large and small areas. Advantages: rapid in action, non-toxic and active in the presence of organic material. Disadvantages: expensive, can cause chemical irritation, corneal damage, should be stored properly in the dark containers. Second is the intermediate level disinfectant. Example is the alcohol. So it can be used in preparations like ethyl alcohol, isopropyl alcohol, most popular alcohol in the hospitals. Action: rapidly bactericidal except spores. Action by denaturation of the proteins. Uses: alcohol mostly used in the concentration of 60 to 80%, can be used as the hand rub. Example, sterilium. Disinfecting smaller non-critical instruments like thermometer, small medical items like vaccine vials or external surfaces of the equipment, stethoscope. Disinfection of non-critical surfaces, lab bench area. Disadvantages: flammable, must be stored in the cool ventilated area, evaporate rapidly, may damage the tips and lenses, inactivated by the organic matter. Second is the phenols. So phenol, carbolic acid, first widely used as an antiseptic by Joseph Lister. Mechanism of action: disruption of cell wall and cell proteins. So there are two uses: used as a disinfectant, example Cresol or Lysol. So it is used as a disinfection of the environmental surfaces, that is bedside tables, bed rails, laboratory surfaces, non-critical medical devices. It is toxic to skin, so not used as a antiseptics. 5% phenol can be used for the disinfection of the sputum specimen as a mycobactericidal. Used as an antiseptic, so it is chloroxylenol. It is a active ingredient of the Dettol. So widely used as an antiseptic. And advantages: it is active in the organic matter. Disadvantages: may cause hyperbilirubinemia in the infants. Third is the halogens. So iodine and chlorine are the examples. They have antimicrobial activity. So first is iodine. Mechanism is disruption of protein and nucleic acid. So there are two preparations: tincture iodine, 2% iodine in potassium iodide, and iodophore, it is povidone iodine. Example is betadine. So it is used as an antiseptic. So 5% topical solution and ointment for wound healing, 7.5% for the hand scrub, 10% for the surgical skin preparation, 1% oral antiseptics or for as a mouthwash, and as a disinfectant for the medical equipment. So it is also used in hydrotherapy of the tanks. So next is the chlorine and hypochlorite. Mechanism of action is the oxidation of cellular materials and destruction of the vegetative bacteria and fungi. So there are two preparations: free chlorine and hypochlorite. Hypochlorite is available in liquid form, that is sodium hypochlorite, or household bleach, and powder form calcium hypochlorite or bleaching powder. So uses of the free chlorine, disinfection of the municipal water supplies and swimming pool water, also in the dairy and food industries. Uses of the sodium hypochlorite, it is available in the concentration of 5 to 6%. It should be used in appropriate dilutions for disinfection of various hospital supplies. Contact time 10 to 20 minutes. Advantages, it is broad spectrum, sporicidal, rapid and non-inflammable and low cost, widely available. Disadvantages, inactivated by the organic matter, toxic to skin, mucosa, carcinogenic. So daily preparation may cause corrosive damages to the fabrics, carpets. Third type is the low level disinfectant. It destroys all the vegetative and enveloped viruses. Other actions is invariable. So example, quaternary ammonium compounds. So mechanism is by producing enzymes, denaturation of the cell proteins and cell membrane. The preparations are the benzyl ammonium chloride, but it is not acting in presence of hard water. But newer generation quaternary ammonium compounds are active in hard water and better compatible. Cleaning agent. So cleaning is important step for the before disinfectants applied to remove organic matter, because few reagents do not act in the presence of organic matter. So the examples are enzymatic cleaners. So they contain enzymes like amylase, lipase and protease and all. Cleaning chemicals, so they are detergents. So manual cleaning and automatic mechanical cleaning like ultrasonic washers, disinfectors. Test for chemical disinfectants to check the efficacy potency of the disinfectants. So the various methods are available. So first is a Riddle-Walker or phenol coefficient test. Second is the Chick-Martin test. Third is the capacity, that is Kelsey-Sykes test. And fourth is the in use, that is Kelsey-Maurer test. Ethylene oxide or ETO sterilizer most commonly used in the CSSD. Mechanism is microbicidal, where it causes disruption of all the cell components, that is cell proteins, DNA and RNA. The sterilization cycle you have to carry out in the 3 stages. So first preconditioning, then sterilization and then aeration. Preconditioning, air is replaced by the vacuum. Sterilization, ATO is allowed in the chamber, so gas concentration, temperature, humidity, exposure time should be noted. Then aeration, that is degassing, so extensive aeration to remove residual ATO, because ATO is a toxic and carcinogenic. The uses used by CSSD to sterilize critical items, sometimes semi-critical items that are moisture or heat sensitive and cannot be sterilized by steam sterilization. So examples, heart-lung machine, components, sutures, catheters, stents, respirators and dental equipment. Advantages, suitable for the heat sensitive items. It has high penetration power, highly diffusible, penetrates areas that cannot be reached by the steam, non-corrosive to plastic, metal and rubber material. Disadvantages, highly inflammable, irritant, explosive and carcinogenic. So ETO is usually supplied in 10 to 20% concentration mixed with inert gases like either CO2. The long duration of cycle 12 to 14 hours. High cost of instrument and consumable. Sterilization control, spores of Bacillus atrophius is the biological indicator. Second is the plasma sterilization. So it is a gaseous state consisting of ions, photons and electrons and free radicals such as O and OH. So it is the most commonly used device in the CSSD to create plasma state. So process, first vacuum is created, then second chemical sterilant, that is H2O2 is vaporized for 50 minutes at low temperature 37 to 44 degrees Celsius. Then gas plasma is produced by the applying electric field. Last step, excess gas is removed. No aeration required because byproducts are not toxic. Cycle duration 75 minutes. Uses by CSSD for sterilization of materials and devices that cannot be tolerating the high temperature and humidity of steam sterilizer. Example, plastics, electrical devices, corrosion susceptible metals such as orthoscope. Precautions or disadvantages. So my item should be dried before loading. Then linen or paper or cellulose liquid cannot be processed, may not penetrate well, especially in channels or devices for long with long lumen, has small chamber, cannot be used for bulky amounts, expensive equipment and packing materials. Sterilization control is the biological indicator, spores of Bacillus stereothermophilus. So these charts suggest the summary of the all disinfectants. So glutaraldehyde, Sidex, aldehyde type 2%, mostly for the sterilization of all the scopes. Povidone iodine, halogen group 2 to 10%. It is used mostly as an antiseptic, wound clinic and mouthwash. Dettol, phenolics group, mostly antiseptic for the wound. Hypochlorite, so it is 1 to 5%, mostly 1% hand washing, 4% disinfectant. Hydrogen peroxide, oxidizing agent 3 to 5%, disinfection of the wound and environmental surfaces, mainly used for the fogging. Formalin aldehyde group 10%, preservation of anatomical and histopathology specimens. Phenol carbolic acid 5%, disinfection of environmental surfaces, floor, walls, disinfection of the sputum, skin, antiseptic.
Hello students, let's study general bacteriology.
Hello students, today we will see about bacterial cell. So components, what are the components? There are coverings of the bacterial cell. So outermost covering is called slime layer and the function of this is protection of the bacteria from antibiotics and disinfectants. Second covering is capsule. So it is gelatinous substance secreted by bacteria which becomes thick to form capsule around the cell wall and function is protection and demonstration by India ink preparation, that is negative staining. An example of capsulated bacteria are pneumococcus and fungi is cryptococcus. Cell wall, it is outer layer of bacteria and function, it gives shape and rigidity to bacteria. Cell membrane, the inner membrane, inside the cell wall there is a thin semi-permeable membrane and function, it controls inflow and outflow of metabolites, it gives mechanical strength to the bacteria. Cytoplasm, it is a liquid and water liquid suspension in which water and organic inorganic substances are there and plus organelles are there like mitochondria which is for the powerhouse of energy, ribosomes for protein synthesis, Golgi complex for protein synthesis, endoplasmic reticulum, so it is a network of cells in near the nucleus for the protein synthesis. Lysosomes, they are sac-like structures present for the destruction of the foreign material. Nucleus, it is long filament of DNA coil inside cytoplasm. As bacteria is prokaryotic, the well-defined nucleus is not there and no nuclear membrane and no nucleolus. Function of nucleus is bacterial division, antibiotic resistance to carry from one cell to another cell. Plasmid, it is a extra genetic material present and it is function as a transfer of genetic material. Thank you. Hello students, now today we will see special structures of bacterial cell. So first is flagella. So flagella in motile bacteria, they possess one or more long unbranched filament that is called as flagella and function is it is organ of motility or locomotion. Demonstration, it can be demonstrated by dark ground microscope or electron microscope or motility testing, that is hanging dropped preparation. Classification of flagella according to the arrangement is peritrichous flagella are all around the cell, monotrichous single flagella at one pole, amphitrichous single flagella at two poles, lophotrichous group of flagella at one pole. And here in this diagram you can see the flagella arrangement. Second structure is fimbria or pili, it is organ of attachment. Next structure is spores, so they are highly resistant, demonstrate state of this bacteria, not destroyed by boiling and they are destroyed by or killed by only autoclaving, temperature 121 degrees Celsius for 15 minutes and 15 lbs pressure. Function, to survive the bacteria in adverse condition. Demonstration by special staining, gram stain, Zn stain. Thank you. Hello students, today we will see bacterial classification depending on the gram staining reaction. So gram positive are purple in color and gram negative pink in color. So cocci gram positive, they are staphylococci, streptococci, pneumococci or enterococci, and gram positive bacilli, they are divided into aerobic and anaerobic. So aerobic are bacillus spore forming and anaerobic spore forming are clostridium. Then gram negative bacteria cocci are Neisseria, and gram negative cocci are gram negative bacilli are divided into different classifications. So one can be according to the lactose fermenting property on Meconium agar, they are lactose fermenting and non-lactose fermenting. So lactose fermenting bacteria are E. coli and Klebsiella, and non-lactose fermenting bacteria are Salmonella, Shigella, Proteus, Vibrio, Pseudomonas, Acinetobacter, etc. Thank you. Hello students, today we will see growth requirement of bacteria. So first is food or nutrients required for the growth of bacteria is proteins, polysaccharides, lipids, nucleic acids, organic, inorganic salts and vitamins. Then moisture is required. Then pH around 7.2 to 7.6. According to that, acidophilic and basophilic bacteria, they are known, classified. Hello students, today we will see bacterial physiology, that is bacterial growth curve. So when bacterium is inoculated into a suitable culture media or liquid media and incubated, its growth follows a definite course, and that curve is called as bacterial growth curve. There are four stages in bacterial growth curve. First is lag phase, LAG. Second is log phase, then stationary or exponential phase, and then log phase or exponential phase, stationary phase and decline phase. In this diagram you can see these four phases. So what is lag phase? So immediately after inoculation on culture medium, there is no appreciate increase in number, but there is increase in size of the cells. So this is called lag phase. Log phase, after the adaptive to the new environment, bacteria multiply, and then it is also called as exponential phase. There is increase in bacterial number, bacterial count. Then stationary phase, after log phase cell division stops because of decrease in nutrients and accumulation of toxic products, and then the bacterial count is static, and then after that bacterial count decreases because of the death of the cells because of the toxic accumulation, and this is called as decline phase. So these are the four phases of the bacterial growth curve. Thank you.
Students, let's study direct diagnosis. So let's study direct diagnosis. So first step is sample collection, transportation and storage, and then processing. So there are some general remarks for handling. So all specimens are collected before treatment. Collect specimen in adequate amount from infection site. Use sterile containers. Use gloves while collection and handling specimen to prevent transmission of infection. Use of mask to prevent transmission of infection. Use of septic cabinets while working with respiratory pathogens. Universal precautions must be followed while procuring and handling specimen to avoid infectious agents. The specimen must be labeled properly, bearing the name of the patient, sex, age, hospital registration number, ward number, etc. It should be accompanied by requisition form bearing the corresponding patient's details along with provisional diagnosis and any specific test to be performed on the sample. Samples are blood, swab up upper respiratory tract, pus, sputum, stool, urine, urethral cervical exudate, CSF, skin scraping, etc. Specimen transport. Most specimens transport time should not exceed 2 hours. However, there are some exceptions. Immediate transport within 15 minutes for CSF and body fluids, ocular specimens, tissue specimens, suprapubic aspirate, bone specimen. Urine added with preservative like boric acid, transported within 2 hours. Stool culture transported within 1 hour. Anaerobic culture transported in Robertson cooked meat broth or any other anaerobic transport system. Storage before processing. Most specimens stored at room temperature up to 24 hours. There are some exceptions. Blood culture incubated at 37 degrees Celsius immediately on the receipt. Sterile body fluids immediately plated on receipt, incubated at 37 degrees Celsius. Corneal scraping plated at bedside. Stool culture stored up to 72 hours at 4 degrees Celsius. Urine midstream or from the catheter. Lower respiratory tract specimen gastric biopsy for Helicobacter pylori stored up to 24 hours. Microscopy, compound microscope principle. A microscope is used to produce an enlarged and well-defined image of objects too small to be seen with naked eyes. The degree of enlargement is called magnification or magnifying power of the microscope. Parts are mechanical parts, base, stand, handle, and stage. Magnifying parts, optical lens system, objectives 4x, 10x, 40x, 100x, and eyepieces. The 10x is low power, 40x is high power, 100x is oil immersion lens, and 4x is the scanner. Illuminating parts, condenser, iris, diaphragm, and mirror. The uses of parts: base gives stability to microscope, stage to keep slide on it, objectives for magnification of object, eyepiece to see magnifying image, condenser to pass light from lamp, iris diaphragm to reduce or increase the amount of light by reducing or increasing the opening, mirror to pass sunlight on object. Types of microscope: simple microscope to see insects, compound microscope to see bacteria, fungi, parasites, electron microscope to see viruses, phase contrast microscope, examination of living material like spirochetes, motility. Diagram microscope, condenser is the important part to see spirochetes. Fluorescent microscope, diagnosis of tuberculosis by staining with the fluorescent lights. Compound microscope principle: rays from light source pass through the iris and fall on the specimen. Light rays are gathered by objective and magnified image is formed. Uses: morphology of bacteria, fungi, cell culture after staining, wet mount preparation for living organisms, motility, serological test. Dark field microscopy. Object appears bright against the dark background by use of special dark field condenser. Central opaque area blocks light from entering the objective lens. Peripheral annular hollow area allows light.
So we already saw the procedure. So that is, first you have to pour crystal violet, wait for 1 minute, then wash, then pour gram iodine, then wait for 1 minute, then wash under tap water, then decolorize for few seconds with acetone alcohol, and then apply saffron in with 1 minute, and then wash and then dry, put oil, and then see under microscope oil immersion lens. Principle of Zn staining is the organisms like Mycobacterium tuberculosis, which causes tuberculosis, and Mycobacterium leprae, which causes leprosy, are acid fast. So that means they are resistant to acid treatment. So that is why it is also called as Zn stain or acid fast stain. So this acid resistant to acid treatment is because of the lipid containing in the cell wall of this mycobacterium, that is mycolic acid, and because of that they are not decolorized by strong acids and alcohols after they bind with the carbolfuchsin, that is primary stain. And then because they are not decolorized, they are not stained by counter stain, they are stained by carbolfuchsin, that is pink in color. That is why in blue color background or other bacteria are blue in color, pus cells, neutrophils are blue in color, and then you can see pink color rods of the mycobacteria, either tuberculosis or leprae, according to the sample. So in tuberculosis it is sputum sample, and in leprosy it is skin scraping. Hello students, today we will see Albert staining, that is done for identification of metachromatic or volutin granules in case of Corynebacterium diphtheria. If the patient is having diphtheria, then you have to take the throat swab. So take throat swab with help of tongue depressor, and then spread it over the smear slide, and then make a smear, then air dry, heat fix, and then after that for the staining of Albert you have to pour Albert stain for 3 minutes, then wash it with the tap water, then flood the slide with Albert iodine for 2 minutes, then wash the slide with tap water, air dry, and observe under eye immersion lens by applying oil on that.
To study motility of bacteria by hanging drop preparation. So it is used to know the motility of the bacteria. So procedure: apply petroleum jelly at four corners of the cover slip, transfer drop of bacterial suspension on the center of the cover slip, then take cavity slide and invert cavity portion over the drop. Press slide by soft hand so that cover slip gets stick to the slide. Then carefully invert the slide preparation so that cover slip at the top and drop is hanging, and then adjust the edge of the drop at low power lens, and then at high power objective confirm the motility. And if the organism is moving, then that means it is positive. The motility test of the organism gives idea of the shape, size, and appearance, and the flagella is there or not there. And then examples of motile bacteria are Vibrio cholerae, which has the darting motility or fast motility. Then other examples are Salmonella, E. coli, Proteus, and Pseudomonas.
So first we will see what is definition of culture media. So culture media is a media required for growth of the microorganisms like bacteria, virus, fungus, parasites, all. So basic, but routinely we use culture media for bacteria and fungi. So now classification of culture media, they are divided into different types. So first is based on consistency. They can be liquid, semi-solid or solid. So in liquid, peptone water, nutrient bath, or in solid blood agar and other all culture medias. Based on oxygen requirement, it can be aerobic medium or anaerobic medium. So the bacteria which grow in presence of oxygen, they are called as aerobic medium, and media which do not provide oxygen for the growth of the anaerobic bacteria like clostridium, which do not grow in presence of oxygen, they are called as anaerobic media. So first ingredient is nutrient broth, which contains peptone, that is protein part, then meat extract, sodium chloride and water. And if you add agar to this nutrient broth, it will become nutrient agar. So role of agar is to solidify the media. The concentration used is 2%. If you use 6% concentration of agar, then it prevents swarming of the bacteria like Proteus. Enriched medium, here blood, serum or eggs are added to the basic media and it is called as enriched media for the nutritional deficient bacteria which grow in presence of the extra added nutrients. So examples are blood agar, chocolate agar, Loeffler serum slope and egg based media, that is Lowenstein Jensen medium. And then if more than one type of bacteria are present, special substances are added to favor the multiplication of particular species of the bacteria or to inhibit the other bacteria. So they are called as selective media. Examples are selenite F broth, tetrathionate broth, alkaline peptone water, bile broth. Selective media, selective agents are added to solid the media and which act as an enrichment, but they are solid in consistency. So they are bisalt agar, deoxycholate citrate agar and TCBS for the Vibrio. Indicator medium, indicator in the media changes color when particular bacterium grows in them. So example of that is blood potassium tellurite agar, Wilson Blair medium. So blood potassium tellurite agar is used differential media which differentiate the colonies from one to another. Example is McConkey agar. So on some delicate bacteria, they need transport media, so it is Stewart's medium or Venkatraman Ramakrishna medium for Vibrio, Carrie Blair medium for Salmonella, Shigella, Vibrio, etc. Anaerobic medium, so the bacteria which do not grow in presence of oxygen, so media provides deficiency of oxygen in that medium and they are called as anaerobic medium. So they are Robertson cook meat media and thioglycolate broth media. Some media are there for the growth of the fungal like Candida, Aspergillus, Penicillium, Rhizopus, Cryptococcus. So they are the ideal culture media for fungal isolation is Sabrot's dextrose agar, and dextrose is 4 gram, neopeptone 1 gram, agar is 1.5 gram percent and distilled water. So here the pH is 5.4 and then because of that other commensals are not grown to this acidic medium. The antibiotic sensitivity testing media is done on Muller-Hinton agar for the antibiotic sensitivity testing, and that is usually done by Kirby-Bayer disk diffusion method. Media. So sticking on the plates is done this way. So it is first you have to take this, you have to heat the wire loop on flame till it becomes red hot. Then wait for some time so it will be cool, and then after that you have to take the sample in wire loop and then do primary inoculation. Then from that primary circle you have to draw the lines horizontal, then rotate the plate, then again draw the lines, then rotate the plate, again draw the lines. So this, and then after that tail in the end. So this is primary, secondary, tertiary and quaternary inoculation you have to do, so that you will get isolated colonies to study colony characteristics. Now we will see biochemical reactions for the conformation of the bacteria. So for gram-positive bacteria, that is Staphylococcus, we have to do catalase and coagulase test. So catalase, you have to take 1 drop of colony suspension, then add H2O2 and then see for the bubbles. So catalase positive in Staphylococcus aureus. Coagulase test can be done by slide or tube method. So you have to take 1 drop of colony suspension, 1 drop of plasma, add and see for the clumps. So positive coagulase test is seen in Staphylococcus aureus. Then biochemical test for the gram-negative bacilli is IMVIC, indole MRVP citrate. So indole, you can see this purple coloring in positive test, that is seen in E. coli. Methyl red, you can see the red coloration that is seen in E. coli. Urease test, you can see yellow broth, the yellow slant converted to the pink slant, that is seen in Proteus. Citrate test, the green slant is converted into the dark blue color, that is seen in Klebsiella. Then oxidase test, it is the purple discoloration, that is seen in Pseudomonas, Neisseria, Vibrio. The antibiotic sensitivity testing media is done on Muller-Hinton agar for the antibiotic sensitivity testing, and that is usually done by Kirby-Bayer disk diffusion method.
Let's study general virology. General characteristics of viruses. So definition, viruses are defined as acellular organisms whose genomes consist of nucleic acid but never both, either DNA or RNA, and which obligately replicate inside the host cells. So characteristics are like obligatory intracellular parasites. They possess either DNA or RNA, never both. They are filtrable, smaller than bacteria, no cellular organization, no cell wall, cell membrane, cell organelle, including ribosomes, lack enzymes necessary for protein and nucleic acid synthesis, multiply by complex process, cannot be grown on artificial cell-free culture media, can be grown on embryonated eggs, tissue and cell culture, not susceptible to antibiotics, susceptible to interferon. Morphology of viruses. Nucleic acid is the central portion of the virus. So nucleic acid may be single or double stranded, circular or linear, segmented or unsegmented. Viruses are classified based on nucleic acid as DNA or RNA viruses. Capsid, the covering of the nucleic acid is called capsid. It is made up of protein subunits, polypeptides called capsomers. The function of capsid protect the nucleic acid core from external environment. In non-enveloped viruses, initiates first step of viral replication, antigenic and specific for each virus. Envelope. So some viruses are enveloped. So envelope surrounding the nucleocapsid, it is lipoprotein in nature. Lipid part derived from the host cell membrane and protein part is virus coded. It is made up of subunits called peplomers. Peplomers project as spikes on the surface of the envelope. The significance of envelope, more susceptible to heat and lipid solvents like ether. It is antigenic, facilitates entry of the virus. More than one kind of papillomas can be seen in some viruses, like influenza virus contains hemagglutinin and neuraminidase papillomas. Most viruses are enveloped, except non-enveloped DNA viruses are parvovirus, adenovirus, papavirus, and non-enveloped RNA viruses, picorna, hepatitis A, and hepatitis E virus. Viral symmetry, three types: helical, example Ebola and rabies virus; icosahedral, example adenovirus and poliovirus; complex viruses, bacteriophage. Different shapes of viruses like spherical, brick shape, which is seen in pox virus; rod shaped, bullet shape, it is seen in the rabies virus; tadpole shape, it is seen in bacteriophage; filamentous, it is seen in Ebola virus. Classification of viruses. So they are according to the nucleic acid, DNA viruses and RNA viruses. So let's see DNA viruses. So first is herpes viridae. So viruses are herpes simplex virus 1, herpes simplex virus 2, varicella zoster virus, Epstein-Barr virus, cytomegalovirus, human herpes virus 6, 7, 8. Hepatnaviridae, hepatitis B virus, parvoviridae, parvovirus B19, papovaviridae, human papilloma virus HPV, JC virus and BK virus, pox viridae, variola, that is smallpox, and molluscum contagiosum virus, adenoviridae, human adenoviruses. So these are DNA viruses. Rhinoviruses, picornaviridae, poliovirus, coxsackie virus, enterovirus, rhinovirus, hepatitis A virus, calciviridae, hepatitis E virus, togaviridae, rubella virus, Western equine encephalitis virus, Western equine encephalitis virus, flaviviridae, yellow fever virus, dengue fever virus, and the hepatitis C virus. Coronaviridae, coronavirus, rhabdoviridae, rabies virus, filoviridae, Marburg and Ebola virus, paramyxoviridae, parainfluenza virus, mumps virus, measles virus, orthomyxoviridae, influenza viruses ABC, Guineaviridae, hantavirus, arenaviride, Lassa fever virus, reoviridae, rotavirus, retroviride, HIV, human immunodeficiency virus. These are RNA viruses. Viral multiplication or replication. So stages, first stage is attachment. So viruses get attached to the host cell membrane by the receptor recognition. Then virus penetrates penetration. Then uncoating. So all the parts of the viruses get uncoated. Then biosynthesis. So there is protein synthesis by transcription, protein synthesis and translation mechanism, final protein synthesis, then maturation. So different parts of the viruses gets assembled and then release either by lysis of the cell, that is lytic cycle, or by budding and release, that is called lysogenic cycle. Pathogenesis of viral infection. So infection takes place on the body surface, then it causes localized infection. Example, rotaviruses, intestinal, influenza respiratory, POTS on skin. Then movement of the virons, lymph node blood causes primary viremia. Then the movement to the possible sites of replication, that is blood vessel, bone marrow, liver or spleen. Then they enter blood and then causes secondary viremia, and then shedding blood or other organs. So shedding blood, that is hepatitis B virus, lungs, salivary gland or kidneys. Example, mumps, measles, cytomegalovirus, brain, rabies, polio, measles, skin, varicella virus, nasal and oral mucosa, measles, varicella and rubella. Lab diagnosis of viral infection. So indications to start antiviral drugs, screening of blood donors for HIV, hepatitis B and hepatitis C, surveillance purpose for outbreak or epidemic investigation, and to start the post-exposure prophylaxis in AIDS or hepatitis B virus infection. So first step is sample collection. So samples like blood, throat swab, nasopharyngeal swab, swab from lesions are taken. Precautions taken. Swab collected in VTM, viral transport media. pH is maintained and organism is viable. Cold chain should be maintained. Temperature 4 to 8 degrees Celsius for 24 hours. For longer duration, minus 20 degrees Celsius freezing is done. Direct demonstration of virus. So first is electron microscopy. So we know size and shape of virus. So rabies bullet shape, rotavirus wheel shape, coronavirus penta, corona shape, papillomas, adenovirus space vehicle shape. Then immunoelectron microscopy, fluorescent microscopy, and light microscopy. So in light microscopy, demonstration of inclusion bodies, like example, rabies virus in dog is negri bodies. So inclusion body, aggregates of virons or viral proteins and other products of viral replication that alter staining property of the cell. Host cell. The characteristic of special viral infections, they have distinct size, shape, location, staining properties by which they can be demonstrated in virus infected cells under the light microscope and help in diagnosis of the viral infection. So types are three: intracytoplasmic, intranuclear, and both. So intracytoplasmic, generally acidophilic and can be seen as pink structures when stained with giemsa or using methylene blue stain. Most pox viruses and Negri bodies seen in rabies virus. Intranuclear are basophilic in nature. Example, Cowdery had classified them into Cowdery type A, that is seen in herpes simplex infection. Both intracytoplasmic and intranuclear inclusion gives the appearance of owl's eye appearance seen in cytomegalovirus. What is serological method? So detection of viral antigen by various formats such as ELISA, direct immunofluorescence, immunochromatography, flow through assays. And that is seen in HIV. Detection of specific antibodies, your diagnostic formats such as ELISA, immunochromatography, flow through assays. Molecular methods related to viral genes, nucleic acid probe for detection of DNA or RNA by hybridization method, PCR for DNA detection by amplification, reverse transcriptase PCR for RNA detection, real-time PCR for DNA quantification, real-time RT-PCR for RNA quantification, that is viral load. Fifth step is the isolation of virus. So first, animal inoculation, but because of the ethical issues, animals are not used. Second, embryonated egg inoculation. So hence, embryonated eggs are used for cultivation of viruses. So parts of the eggs are chorioallantoic membrane for pox virus, allantoic cavity, influenza vaccine and yellow fever vaccine, amniotic cavity, isolation of influenza virus, and yolk sac, arboviruses, rickettsia, chlamydia. Third is tissue culture, organ culture. Organ can be used. Then explant culture, that is parts of the organ, and cell line culture. And types are primary, secondary, continuous. In continuous, malignant cells are used so that they divide continuously. Then the growth is seen by uh, cytopathic effects. So these are morphological changes produced by virus in the cell line detected by light microscope. Example, syncytium or multinucleated giant cell formation seen in measles, diffuse rounding and ballooning of cells seen in HSV, herpes simplex virus. Let's study general mycology. Fungi are eukaryotic. They have rigid cell walls containing chitin, mannan, and other polysaccharides. Their cytoplasmic membrane contains sterols. They possess two nuclei with nuclear membrane. They divide asexually, sexually, or by both, maybe unicellular or multicellular. The classifications are of three types: morphological, taxonomical, and pathogenic. So first, morphological classification. So in that yeast is there. Example, Cryptococcus, yeast-like cells. They are oval or round cells with buds. Example, Candida. Molds, so they have hypae, elongated structures. Example, dermatophytes, Aspergillus, Rhizopus, Mucor, and Penicillium. Dimorphic fungi, so they have two forms at the 37 degrees Celsius and at the room temperature. So examples, Blastomyces dermatidis, Paracoccidiioides brasiliensis, Coccidiioides imitis, Histoplasma capsulatum, Sporothrix schenki, and Penicillium marlefi. Second, taxonomical classification based on sporulation. So zygomycetes, sexual zygospores with sporangium within sporangium, asexual sporangiospores, they are endogenous. Example, mucor. Ascomycetes, sexual ascospores within the ascus, asexual conidia, exogenous. Aspergillus basidiomycetes, sexual basidospores in the basidium, asexual conidia, exogenous. Example, Cryptococcus. Deuteromycetes fungi imperfecti, sexual spores not known, asexual conidia, exogenous. Most clinically important spores belong to this group. Based on the infection, that is pathogenic classification. So there are superficial mycosis, superficial surfaces of the body. Example, dermatophytosis. Subcutaneous mycosis, subcutaneous tissue, dermis and subcutaneous tissue. Example, mycetoma, rhinosporidiosis. Deep fungal infections, disseminated. Example, histoplasmosis, blastomycosis. Opportunistic fungal infection, when the immunity is lowered, then fungal infections are caused. They are example, candidiasis, aspergillosis, zygomycosis, mucormycosis, cryptococcosis. Pathogenesis of fungal infection, source of fungus, environment, warm, humid area, air, plants and soil, animals, unsterile instruments and hospital equipments. Site of infection, CNS, eyes, lungs, bloodstream, skin, subcutaneous tissue. The fungal agents according to the sites of infection causes disease. So Cryptococcus is CNS, eyes, rhinoorbital mucormycosis, skin, dermatophytosis, lungs, aspergillosis, bloodstream, disseminated candida, coccidioides, paracoccidioides, systemic and mucocutaneous, subcutaneous histoplasma, mycetoma. Laboratory methods for isolating and identifying clinically important fungi. So first, specimen collection. Second, direct demonstration of fungal elements by microscopy. Third, isolation of the fungus in culture. Four, immunological evidence of infection. Five, other tests for the diagnosis of fungal infection. Example, skin test for hypersensitivity, molecular methods. First is specimen collection. So according to the site, superficial infection, skin scraping, then hair pluck from the root and nails clippings. Subcutaneous tissue biopsy, mycetoma granules from the infection or sinus tract, deep and opportunistic infection, blood, CSF, pus discharge, sputum and other body fluids and aspiration. Microscopy, second step, the potassium hydroxide preparation 10%. It digests keratin. So KOH wet mount can be done for demonstration of fungi. Second, gram staining. In that they are gram positive is cells can be seen. Example, Candida. Cauliflower white stain. Then India ink or nigrosin staining for capsule demonstration of the Cryptococcus. Methenamine silver stain and periodic acid shift stain can be seen in histopath preparation. Lactophenol cotton glue. LPCB can be seen for the demonstration of the colonies. Third step is culture, temperature ranging from 25 to 30 degrees Celsius, slow growing, and then media used taprots, dextrose agar, most common fungal culture media. It contains dextrose for nutrition to fungi and acidic pH to inhibit commensals. Second special media, selective media is bird seed agar, niger seed agar for Cryptococcus, cornmeal agar for the chromogen candida, blood agar, brain heart infusion agar. In preparation of slides, LPCP mounts, so taste mounts in that colony can be taken on slide and then cover slip is applied. But here the morphology of the fungi get distorted. Second slide culture. So on slide, the STA block is taken and on that the growth is done and then can be seen under microscope. Cellophane tape method. So the cellophane tape is applied on the agar block and then can be put on the slide and then growth is seen, colony is seen. So that is elephant tape mount. Laboratory methods for identifying fungi. So morphological identification, rate of growth at 37 degree or room temperature 25 degrees Celsius. Color and morphology of the colony and its texture. Pigmentation on the obverse, that is front, and reverse, that is backside, whether diffusible or non-diffusible. Microscopic identification. So LPCB mount is done, that is lactophenol cotton glue. So in that lactic acid, phenol and cotton glue for the glue coloration. So in that can see hyphae that are septate or aseptate. So aseptate hyphae is in mucor, septate hyphae is in dermatophytes. Mycelium, vegetative or aerial, conidia or sporangium. Then other tests for identification of fungi, germ tube test in Candida, hair perforation test in dermatophytes, urease test, Cryptococcus, fermentation test, antifungal susceptibility test, antibody detection, antigen detection, cryptococcal antigen in CSF by latex agglutination, rapid diagnostic test, nucleic acid hybridization, polymerase chain reaction test. Let's study general parasitology. So classification of parasites, two major groups, protozoa and helminths. So protozoa can be amoebae, flagellates, sporozoa. So protozoa unicellular, so amoeba, Entamoeba histolytica, Naegleria, Acanthamoeba. Flagellates like Giardia, Trichomonas, vaginalis. Blood parasites like Leishmania, Trypanosoma. Then sporozoa, that is Plasmodium babicea. Other like Toxoplasma, Isospora, Cryptosporidium and Melantidium. Helminths are multicellular. So first is platyhelminths. So they are flat and segmented. Cystodes tape-like. Example, Tinea, Hymenoleupisthana, Diphyllobothrium and Echinococcus. Trematodes leaf-like, unsegmented. So cystosoma. Nemathelminths, nematodes cylindrical and unsegmented. Intestinal nematodes: Ascaris, Ankylostoma, Trichuris, Enterobius, Trichinella, Strongyloids. And tissue nematodes: Wucheria and Dracunculus. Loa loa. Lab diagnosis. So first specimen can be stool or rectal swab, then demonstration of parasitic forms by microscopy, culture, animal inoculation, xenodiagnosis, immunodiagnosis, that is skin test, serological test, molecular diagnosis, DNA or PCR, other laboratory tests to detect cytological or biochemical alterations in blood like CBC, IgE.
Now let's study indications of examination of stool. So first it is required for the detection of parasites, infestation of intestine like hookworm, roundworm, Entabima histolytica, which causes amoebic dysentery. Then bacterial infection of intestine studied by cultural examination of stool like bacillary dysentery, which caused by Shigella, enteric fever, cholera. Then chemical examination of stool is done to detect defect in absorption, that is malabsorption syndrome. Urobilinogen estimation to study excessive hemolysis. Occult blood test is done for detection of ulcerative lesions of upper gastrointestinal tract. For lab diagnosis of parasitic infection, the specimen is stool and it is done for detection of ova, cysts, tropozoites, eggs, larvae of the parasites. So collection of sample is done by either direct stool examination or other samples can be NIH swab, that is National Institute of Health, is used to collect samples from the perianal area in the infection of enterobius infection. Then cellophane tape is applied to the perianal area of the patient early in the morning. Or string test for duodenal samples for tropozoites of Giardia lamblia. So these are different methods of the sample collection of the stool. Then after that, stool preservation can be done in polyvinyl alcohol, merthiolate iodine, Shodin fixative or formalin solution. So this is a stool container. In that plastic container, sterile container with the spoon attached to the lid of the container. Now when to examine the stool sample? So if consistency is liquid stool, then you have to examine it within 30 minutes, because in that we are examining tropozoites, that is motility of the parasites. So tropozoites like amoeba or giardia. Then semi-solid stool we can examine within 1 hour, and solid stools of formed stools up to 24 hours after collection, because in that we see mainly cyst forms. In semi-solid we can see both tropozoite and cyst form. The first step is macroscopic examination of stool. So consistency of stool, like it is liquid, semi-solid or solid. Then blood or mucus present or absent. Then few parts of the intestinal parasites like adult segments like Ascaris, Enterobius vermicularis, tapeworm. So we can see that parts of the parasites. In microscopic examination of stool, first step is the routine examination, that is by saline wet mount and iodine wet mount. So in saline mount, procedure is take 1 drop of normal saline on slide, mix stool sample, put cover slip and see under microscope. The advantages of saline mount: useful in detection of tropozoite cyst of protozoa, eggs and larvae of helminths. Motility of tropozoites and larvae can be appreciated. Bile staining property, that is they appear golden brown, and non-bile stain eggs appear colorless. So non-bile stain eggs examples are hookworm, enterobius and H. nana, HEN. Then iodine mount, the procedure: take 1 drop of Lugon's iodine on slide, mix stool sample, put cover slip and see under microscope. Advantages: the nuclear details of the protozoan cyst, helminth eggs and larvae are better visualized. Disadvantages: iodine immobilizes and kills the parasites, so motility of the tropozoites and helminth larvae are not appreciated, and bile staining property is not appreciated. Second method is the permanent stain smears. So it is done for detection of protozoan cyst and tropozoites by staining their internal structures. The common methods are: first is the iron hematoxylin stain or trichrome stain and modified acid fast stain. It is useful for the coccidian parasites like Cryptosporidium and Isospora. So in that 0.5% acid, that is H2SO4 is used, and that is why it is called modified acid fastane. And these parasites are mostly opportunistic infections in the HIV patient. When eggs or larvae or parasites are very few in number, then concentration techniques are used. So first is sedimentation method and second is flotation method. So in sedimentation method, the parasites are recovered in the sediment using either gravity or centrifugation. So the techniques used are formalin ether or formalin acetone. So stool sample and these reagents are taken in centrifuge tube, and then it is centrifuged, and then deposit is taken. In that we can find the morphology of the parasites. The advantages of sedimentation method is easy and minimal technical error is there. Morphology of eggs and cysts are preserved and it is more sensitive. The flotation techniques, stool is mixed with solution having high specific gravity, and the techniques used are saturated salt solution or zinc sulfate flotation method or sugar flotation method or saturated magnesium sulfate solution techniques. The disadvantages, some helminth eggs like trematodes, tinea, unfertilized eggs of ascaris do not float in the salt solution. And then distortion of morphology of the eggs and cyst is also there. So in this stool microscopic examination, we can see different morphological forms of the parasites. This is stool microscopic examination. So in that we can see different morphological forms. So first is cyst of Entabima histolytica. In that we can see 4 nucleus. Then cyst of Giardia lamblia. In that we can see 4 nucleus, but the cyst is usually oval. Then tropozoite of the lamblia. So in that we can see flagellas. Then egg of H. nana. In that we can see the circular egg and it with the flagella. Then in tinea we can see it is circular usually, and we can see 3 pairs of hooklets and the transverse striations. Then egg of hookworm, in that we can see 4 blastomers. Then fertilized egg of Ascaris lumbricoides. So here you can see space between the covering, and then the center is the embryo. Unfertilized egg of Ascaris lumbricoides. So here you can see this no space and it is full of the dots and covering. Then egg of vermicularis, Enterobius vermicularis, it is mostly by one side convex and one side flat, and then here you can see in the center embryo. The egg of Trichuris trichuria, so it is usually embryo inside and then covering of double layer and 2 mucus plugs at the both ends.
Let's study bacterial genetics. So, introduction. Genetics is the study of heredity and variation, seeking to understand the causes of resemblance and differences between parents and their progeny. So, in 1940, principle of genetics applied, and that led to birth of new branch of science, that is molecular biology. The principles of bacterial genetics deals with the study of heredity and gene variations seen in bacteria. All the hereditary characteristics are encoded in DNA, present chromosomal as well as extra chromosomal. The mechanisms of genetic variations, mutation, and the horizontal or the transfer or exchange of genetic material is the transformation, transduction, conjugation, lysogenic variable conversion, and transposition. So let's say mutation. So mutation is random, undirected heritable variations caused by alteration in nucleotide sequence. So types of mutations are spontaneous and induced. So spontaneous mutation occur naturally in any dividing cells without adding any mutation. Induced mutation result of exposure of organism to a mutation. Example, physical agent, ultraviolet radiation, cytosine and thymine are more vulnerable to UV rays. Chemical agents, alkylating agents, 5-bromouracil. First type in gene transfer is transformation. So it is transfer of portion of genetic information through the free DNA. So in that Griffith experiment is there stated that the live non-capsulated strains were transformed into capsulated strains due to transfer of capsular genes released from lysis of the killed capsulated strains. So second is a transduction, transfer of portion of DNA by bacteriophage. So there are two types: generalized transduction. Any segment of donor DNA is transferred at the random. Restricted transduction, specific bacteriophage transduces only a particular trait. Example, lambda phage of the E. coli. So in this you can see bacteriophage. So phage DNA enters inside the host bacterium, then destruction of host DNA, synthesis of virus DNA and code proteins, virus capsid synthesis and virus assembly, then transducing particle, cell lysis, phages infect another cell, survival of donor DNA. So integration of donor DNA into recipient chromosome or degradation, but so that is unstable gene transfer or unstable gene transfer or the abortive transduction. Lysogenic conversion, bacteriophage, two types of life cycles, virulent, that is lytic cycle, progeny phages inside the host bacterium, ruptures and release. Temperate or non-lytic cycles, that is lysogenic cycle, whose bacterium unharmed phage DNA integrated in bacterial chromosome as prophage multiplies synchronously with host DNA. Example, C. bacterium. So phage-coded toxins are diphtheria toxin, cholera toxin, virocytotoxin of E. coli, streptococcus pyrogenic exotoxin, and botulinum toxin C and D. The fourth is the conjugation. So male or donor bacterium, physical contact with female or recipient bacterium leads to transfer of genetic elements through the conjugation tube. So bacterial conjugation and sex pillars or conjugation tube. E. coli, K2, K12 role of plasmid in conjugation. First recognize plasmid sex factor or fertility F factor. So here you can see donor factor. So that is donor DNA, and then recipient DNA is there. So F plus is donor DNA, F minus is recipient DNA. So pilus connects cells, conjugation tube is formed, and F factor is replicated and transferred to the another recipient DNA. The fourth stage is the transposition. Transposons or transposable elements are the bacterial genes that are capable of intracellular transfer between chromosome to chromosome, plasmid to plasmid, chromosome to plasmid, and vice versa. Process of such intracellular transfer is called transposition. Gene transfer by artificial methods. So it is called genetic engineering. It is referring to deliberate modification of an organism's genetic information by directly altering its nucleic acid genome. And the mechanism is known as recombinant DNA technology. The applications of the genetic engineering: production of vaccines, production of antigens used in diagnostic kits, production of proteins used in therapy, transgenic animals, gene therapy. Blotting techniques are southern blot to detect DNA, northern blot to detect RNA, western blot to detect proteins, eastern blot, it is modification of western blot, analyze proteins for post-translational modifications using probes that may detect its carbohydrate and phosphorylation. Difference between mutational resistance and transferable resistance. So mutational resistance involves resistance to one drug at a time, transferable multiple drugs. Mutational resistance, degree of resistance usually low, and transferable it is high. Mutational resistance may be overcome by high drug doses, transferable ineffective. Mutational can be prevented by combination of drugs, transferable cannot prevent. Mutational resistance does not spread, transferable spreads to other cells. Mutational resistant mutants are usually metabolically defective, and in transferable they are normal. Mutational resistance virulence may be low, transferable virulence not decreased.
Hope you find this session useful for your understanding and for your exams. Thank you.